Good morning, and sorry for the slight delay. So I'm just going to start the day with a talk describing some of my experiences a few years ago with an outbreak that happened in Germany. And the idea here is just to provide context to the work you're doing in a way to change your attitudes to things and make you think more broadly about how various methodologies and approaches play out in the real world and how they're going to play out in the future and how things change. So the key story of this particular outbreak was that we broke a lot of paradigms. It changed the way people thought about things, and we all of us carry around, often unwittingly, these assumptions in our head about the way the world is. And we have to come to terms with the fact that sometimes the world isn't quite as we expected. So here's a number of assumptions or paradigms. I mean, if you want to get purist, are they all paradigms? They're assumptions as much as paradigms. That were perhaps in the heads of most microbiologists and members of the public as well, perhaps, before this outbreak. So paradigm one, infection is not a threat to advanced societies. Well, obviously, now we're over the other side of still in a COVID pandemic. Clearly, we don't accept that anymore. But if you go back 10 years, people were saying that kind of stuff. Oh, if you're in a medical school, the medical schools should be concentrating on heart disease, cancer, infection, that's a done deal, just forget it. Bacterial genome sequencing, slow, expensive, onerous task, as I mentioned in earlier talks. In the beginning, it was like almost like an Apollo mission, costing you hundreds of thousands or millions of pounds, and it's hard to shake off the idea that it's sequencing genomes is a slow process. Serious science, well, serious science is writing papers and submitting them to journals and getting them peer reviewed. And that's all done behind closed doors. And the rest of the world sees about your research is when it appears in a peer-reviewed journal. That's the way people used to think. Also, this idea that pathogens can be tidied up into nice groups called pathotypes, where we can say, right, that's what that is. And this is this idea of this kind of typological thinking, which we spoke about when we spoke about evolution, the idea that everything can be tidied up. Another idea is that you have to grow pathogens to detect and characterize them, and that you have to get those colonies on the agar plates before you can make any diagnosis or characterization. And then finally, another paradigm that we're going to at least explore overturning is the idea, this is a quote from, I think it's from Hamlet, when troubles come, they come not single spies, but in battalions. We'd assumed up till then that when pathogens cause disease, they just do it on their own, one pathogen, one disease. Maybe it's not that simple in life. So just to provide some context, this is the way people were thinking back then about E. coli. So E. coli, it's one of the most important organisms in biology. It's the most important model organism. It's a commensal that's found in most of the people in this room. There's maybe one of you that may be not carrying E. coli, but the rest of us will be. But it's also an important cause of disease. And it causes disease in the gut, and it can escape from the gut and cause urinary tract infections, bloodstream infections. But if you're trying to pin down the different kinds of E. coli that cause disease in the gut, this is one table where people are trying to tie it down and say, right, this is what the pathotype is. These are the defining markers of that pathotype and essential virulence determinants. And you can see, when you look at where those essential virulence determinants are, they're often on mobile genetic elements, pathogens, the islands, plasmids, transposons, and so on, prophages. So that might lead you to think, well, is it going to be clear-cut then? Because if things are actually moving around all over the place, there's going to be plenty of scope for blurring those distinctions and those boundaries. And here's just another thing that shows you the same kind of thing in a graphical format, if you like. So we've got the enteropathogenic E. coli up the top there, where they stick around and manipulate the actin cytoskeleton and form little pedestals. EHEC does the same, it was thought, and had to have this particular Lee-encoded type 3 secretion system, but had this particularly nasty toxin on top of that as well. ETEC had various kinds of toxins, not quite so nasty, but tended to cause diarrheal disease nonetheless. Enteroaggregative E. coli, the main thing was it was kind of forming biofilms and sticking in a certain way, but also putting in toxins that cause disease. Enteroinvasive E. coli is effectively a variation on Shigella. It's effectively lactose fermenting Shigellas, and it manipulates the actin cytoskeleton, invades cells, lives inside cells, can move from one cell to another. And then diffusely adherent was another one that kind of extra also ran in sense of the main pathovars. That's just to provide some context. Now, I'm doing this in the style of a story, and our story begins in 2011 on this blog post here saying that I'd happened to win a new instrument called an ion torrent. So this was at a time when the so-called next generation, which is a bit of a misnomer, kind of high throughput sequencing platforms had come to market and were making inroads. But the biggest change was that we actually had these desktop instruments that had a kind of form factor that just like the size of a photocopier or even the size of a printer. You know, they'd sit on a desk or a bench, and they would fit in a normal lab. You know, the average microbiology lab, whether it's a clinical lab or a diagnostic lab or a research lab. And so Ion Torrent, who had come up with this new instrument, they had launched in the US, but they'd also then launched a competition in Europe saying, oh, we're going to give away a couple of these instruments to the person who can come up with the best application. So I put in, we're already sequencing Acinetobacters and tracking them in outbreaks. If we had one of these, we could do that on speed even more. And they took my Blarney and they said, all right. And so I went out to Lausanne in Switzerland, and they wined and dined us, and I got given an ion torrent. So the thing about the ion torrent was it's so new that nobody knew how to analyse the data from it. That's where we came into our own, if you like. So just around the same time, a few weeks later, this outbreak struck. So it's an outbreak caused by a particular serotype of E. coli called O104H4. And it struck in May and June of 2011. There were over 5,000 cases across Germany, and you can see that most of them are in the north of Germany, but they were scattered across Germany. And there were over 50 deaths. And the thing that was particularly striking, I guess you'd say particularly tragic about this really, was that those deaths were in people who were young, often, and there were people who were otherwise well. And they were suddenly struck down with this and became critically ill and died. It took a bit of a time to work out what was the vehicle for this. Initially, there was an idea it might be cucumbers, and that caused a lot of problems, I think, and bananas, actually. No, it was bananas, not cucumbers. And that led to a lot of problems in terms of the price of those, and the fell and so forth. But it was finally linked to sprouting seeds, bean sprouts, in a salad. And one of the things that was particularly striking about this outbreak was there was a high risk of this severe consequence of E. coli infection called hemolytic uremic syndrome. So when the sugar-like toxin is released, it gets into the bloodstream, it causes the destruction of red blood cells and also damages the kidneys. So the other thing was that females are particularly at risk in this outbreak for reasons that are unclear. So this immediately knocked out the idea that infection doesn't matter. Here was one of the most advanced societies in the planet having to deal with a serious and very difficult to handle outbreak that was actually killing people. This shows you the epidemic curve of the outbreak, and you can see there that there is this predominance of infection and also of the most severe consequence, hemolytic uremic syndrome, in females. And I haven't seen a good explanation of why that is. If you want to be trite and silly, you can say, real men don't eat salad, they don't eat bean sprouts. But I don't know if that's the explanation in terms of whether it really was that more women were exposed or whether there was some other reason, but it's not quite clear. This is the guy in Hamburg. He was a clinical microbiologist, and he was at the centre of having to deal with this outbreak. And he was trying to make sense of the strain that was causing the disease. But he didn't have, within his own clinical lab, the wherewithal to sequence genomes. This was still, this is over 10 years ago. Those desktop instruments had just arrived on the scene. Now, I'm showing my age here by making a bit of a joke. I don't know if this means anything. When I was growing up, there was Thunderbirds. There was a TV series, and if you were stuck in some difficult situation, you could call international rescue, and some satellite would wake up, and they would send the various Thunderbirds to come and save you. Does anyone else recognise the illusions? They did make. It was rerun in recent years as well. So he called international rescue effectively. Through a contact, a Chinese person working in the lab said, well, the Beijing Genomics Institute could help you with the genome sequencing. And so, he called Thunderbirds I'll go, and it didn't quite happen in Thunderbird 2, but the DNA was sent to BGI, the Beijing Genomics Institute, which was based in Shenzhen, just over the border in the People's Republic of China, just over the way from Hong Kong. And they had just taken receipt of their first iron tolerant instrument as well. And so they thought, well, let's have a go. We'll run it through the iron tolerant and see what we get. Now, the thing that was quite remarkable here was that they sequenced it in China, and they immediately released the sequence data into the public domain. So I'm not sure if you're familiar with this idea of Creative Commons. You come across this as an idea. So instead of asserting your copyright, you can just say, I'm just releasing this into the public domain under and there are a variety of licenses available under the Creative Commons initiative, where you can say, I'm just releasing this and anyone can do whatever the hell they like with it, which is what they did here. They released it under CC0, which basically meant we're taking ownership of this, you do what you like, international community, people out there. More commonly, people they use a Creative Commons license, but they say things like, well, you've got to provide an attribution if you do anything and release this further. You have to say that I did this work. And, okay, you did downstream value added, but you have to recognise me when you release it again. Sometimes they say that you can't use it for commercial purposes and so forth. But basically, there was an old saying on the internet, which is that information wants to be free. From the early days of the internet, the idea that just let the information get out there, and this was quite revolutionary in its way. It goes against the kind of undergoing assumptions that science is something you do very secretly in your lab, and you're looking over your shoulder. You might get scooped if someone knew what you were doing or whatever. So hats off to these guys for actually releasing the data. Now, at that time, Nick Lohman was working for me. He was doing a job of work and also studying for a PhD. And he had just got into Twitter and into use of social media. And he'd also been teaching himself how to handle the data coming off the ion torrent. So he was one of the few people in the world who had access to an ion torrent and was thinking about what does the data look like, how do you do base calling and so forth. And so he said, oh look, they've released 5 runs. It's released without any restrictions. He happened to be at a bioinformatics conference, the Applied Bioinformatics and Public Health Conference in Cambridge. And what he did was he put up a first assembly of those reads that had come out of the ion torrent. You can see it was a pretty rubbish assembly with 3,000 contigs, but it was nonetheless total base is 5.4 million. It was in terms of quality, it was okay. In terms of it wasn't contaminated. It was a reasonable thing. And so he released his assembly into the public domain and said, "Yeah, rest of the world, do what you like with this." Effectively invited the bioinformatics community to crowdsource the analysis of the genome. So he had, within 24 hours of its release, he'd assembled the genome. Within 2 days, others had said, this is actually related to things that are already out there. It's not like the Andromeda strain that's come out from outer space or something. It is related to other E. coli that are out there. Within 5 days, people had said, I've got here's a number of markers that you could use if you want to do PCR to rapidly identify this outbreak strain. And within a week, there were over 24 reports on an open source wiki, which documented the biology, gave insights into the potential virulence factors in there, how the strain had evolved. And this was quite a game changer. Really. We hadn't seen anything quite so like this before. Now, you're probably aware that actually this kind of thing did go on during the COVID pandemic. And in a way, this was the precursor. We set the ground, we prepared the ground for what happened with subsequent outbreaks. And in many ways, this was a change of attitude as much as anything else. It didn't require people to have sophisticated new ways of working. It was just an attitude change that, we worked together. And I mentioned earlier in the week that one of the things about the way bioinformatics works is this idea of altruism, that people just work for nothing. They release the work they've done for nothing to the rest of the community, and everyone benefits. And this was another example. These were people who were not public health microbiologists whose job it was to analyse genomes and work out what was going on in an outbreak. They were people just like you. They were scientists who dabbled in microbial bioinformatics, and they added value to this initiative like that. So here's just to show you this is all happening both on a kind of blog, but also on Twitter as well. When people were doing stuff, they released it. And you can just about see, it's not a great screen dump, but you can see lots and lots of comments there where people have done stuff. But also it was an international venture. There were people, you can see just by looking at the names there, you've got names that are obviously English, but you've got names that are Spanish, you've got names that are Polish, you've got people, in fact we had people from Europe, America, Australia, all chipping in together to actually help analyse this particular strain. Now, while that was going on, this was the big boss in Hamburg, and he was calling international rescue. So I'd actually contacted the group in Hamburg when the outbreak was starting to kick off, and I said, look, we know quite a bit about E. coli, we know quite a lot about genomics. If you need any help from us, this guy, Martin Epfelbacher, he said, no, I think we're okay. We're working with BGI, we'll be all right. Then he actually called me at about 10:30. I think it was a Tuesday, a week or two later, And he said, oh, I'd like to speak to you at midday. I said, well, you can speak to me now. He said, no, I haven't got time, but can I book a chance to speak to you at midday? All right. So at midday he spoke to me, he said, well, we've tried to analyse this outbreak strain. We've written it up. The work was done by BGI. We're working with those guys. We've submitted it to the New England Journal of Medicine. Okay. The commissioning editor seems to be quite interested, but the reviewers, they don't like the way we've done the analyses. Okay. And they don't like the way we've written it up. Okay, so they haven't rejected it, but they don't like the way you've done the analysis or the way you've written it up. Yeah, could you help us with that? Yeah, I like writing things. And I've got this guy, Nick Lohman, who could help with the analyses. He said, there's a snag. It was a Tuesday, actually. I said, why? He said, we've got to submit our revised version by 10 p.m. on Thursday. 5 p.m. US Eastern time. So we've got 48 hours just over to redo all the analyses and write a paper. Yeah. Okay. I accept the challenge. I put the phone down. It was like, what on earth are we doing here? So I sat down with Nick and I said, well, you've got all these tweets and blog posts. These don't really. I'd been on holiday when he kicked that off. Actually, I came back. I said, "Look, this isn't scholarship. You're having a bit of fun on social media. We need to turn this into a peer-reviewed research publication. This opportunity has just landed in our lap. Do you accept the challenge of going away and redoing the analysis? We cannot take the Chinese analysis. The Chinese seem to think that they could get a contiguous chromosome, and we just said." That is simply not possible. It is. There's only one seat, I'm afraid. That's simply not possible from short-read sequencing to get a contiguous chromosome. So we're going to have to politely disagree with them. We won't try and replicate their analysis. We'll just do our own analysis and take what's in the public domain as well. And so we said, right, we're going to have to make it as kind of story. We'll write it up as a case study of one particular family. We'll look at how the analyses took place, and we'll mention the crowdsourced analyses and describe the journey to getting the analyses done rather than just a destination. And Nick took it on his shoulders and went away, did all the analyses and came back. Now we had a problem there because the crowdsourced analyses that we were sort of scooping up, there were dozens of people on there. There were already dozens of people on the draft of the paper, and it was a big question of, do we include the whole consortium or do we include a few or what? In the end, we decided to scoop up some of them. We put them into this E. coli genome analysis crowdsourcing consortium and made that the author, and then had a little note to say who was in that consortium. I went in as a senior author. I said I made that as a precondition that if we were going to do this. And Nick Lohman was also starred. It doesn't show it there, but he was starred as one of the first authors of the paper. So, give you a couple of German phrases. It's a new Weltanschauung, a new view of the world, a new zeitgeist that we brought in here. So we'd shown through this initiative, and Nick had done most of the work. I was the one that actually drafted it all, wrote it all up as a paper, but he was the one that ploughed the furrow, really. Basically showed that, things like blogging and Twitter were not just, time-wasting activities, but they can actually augment the usual channels of academic discourse and prove useful in this setting. And also the fact we'd done this open source genomics, where we'd had this confluence of high throughput genomics, crowdsourced analysis, and this liberal approach to data release. The other key point was there was a key role for benchtop sequencers in this initiative as well. So it was groundbreaking in several different ways. And like I mentioned earlier, it laid the groundwork for the way in which people worked with future public health emergencies. And I did this in one of their podcasts, and Nabil commented on this. What is it? You were doing your PhD at the time. You said, "Oh wow, this was amazing to see all this in real life and all that." It did have quite an influence, I think. The other thing, though, when we actually look at the analyses and the results of the analyses, well, there are some other takeaway messages that were really a bit surprising. So pathogens don't bother with passports. Yeah, I think we kind of got that idea from COVID, haven't we? But at the time, it was like, this is a new strain. In fact, something had been seen similar in Germany 10 years before, and there'd been something seen similar in Korea. In fact, the closest genome sequence strain, though, was from the Central African Republic. And it had come from what had been described as the enteroagerative lineage. But what we found was that it wasn't as simple as that. There was a great challenge here to typological thinking, because it turned out that really these pathotypes, as they had been defined, could overlap and evolve. There's virulence factors in one E. coli. There's no reason why they can't just jump from one lineage to another. So what we found here was that this was actually, although it was an enteroagerative lineage, it was also a Shiga toxin-producing E. coli. But unlike the assumptions up to then about enterohemorrhagic E. coli as a form of Shiga toxin, where we'd always said, they need to have this particular type 3 secretion system encoded by the Li, this organism did without it, didn't need it at all. And the other rather sinister thing was that this was a diarrheal gut organism, but it actually carried antibiotic resistance determinants, even though nobody was treating diarrhea with antibiotics, not beta-lactams, certainly the ones we see here. So we saw all of these things that challenged the way we thought about the world. So that was one interesting little angle that we came out of this work. As I mentioned, that this was the time of great innovation. These 3 benchtop sequences all came to market, roughly around the same time. There was one called the 454 Junior, which was based on an existing technology but just had a much smaller form factor, nice small thing that could fit on the bench. There was the Iron Torrent, which they tried to make all sexy by putting all these silly symbols on the front of it and making it, you could charge up your iPhone on it as well. And then the MiSeq, which was the Illumina version. So this was what they called Selexa sequencing initially, bought out by Illumina. They'd had instruments that were sort of substantial inside, but this was the MiSeq, the first, if you like, sequencer for the masses. And I think I mentioned this before, that Nick Lohmann, we had a kind of plan B. If that relationship with the German team in Hamburg hadn't worked out, we'd already struck up a potential collaboration with John Wayne, who at that time was working in Public Health England. He now works here. And we said what we could do is, we've got a British isolate from that outbreak, someone who'd been in Germany and returned. Why didn't we just run that through the 3 different instruments? And they had a 454 at Public Health England. We had the Iron Torrent. And we spoke to Illumina, and it turned out we spoke to people at Illumina. The person that actually did the work at Illumina was a guy called Josh Quick, who later went to work with Nick Lohmann, and that's another story that you'll hear later about that. And Josh Quick will be coming to see you all in February, I think we've decided. No, in March he's coming. But anyway, at the time, we basically ran it through all 3 instruments, and what we showed was that both the 454 and the Iron Torrent had a similar problem with highly inaccurate results when you looked at homopolymers, whereas the MiSeq coped with those much more easily. So the MiSeq was the more accurate instrument. And this became one of my most highly cited papers. It's my second most highly cited papers. One of my colleagues, who was an FRS, when we were talking about do we put it into the ref, this time when we're judging the quality of research, he was rather dismissive. He said, this is just a witch report on sequencers. It's not bloody science, is it? But nonetheless, we influenced the field, and as I say, it's been highly cited. It was so highly cited, the Illumina reps used to give it out when they were trying to sell people MiSeqs. They'd say, look at this paper, it validates the use of our platform.
Hopefully you got a cut out of it.
I didn't get a cut out of it. I did think, well, I should have bought shares. What did happen was that the iron tolerant people never spoke to us again, and the iron tolerant never got used again. It just sat languishing. And I managed to Illumina said, oh, if you trade it in, we'll give you another MySeq at a reduced rate. And the hospital wanted one, so I said, okay, we could trade it in, but that doesn't feel like the right look. You know, they gave it to me. You know, they might say I'm being dishonourable, or even though I've just done perfectly good science. You know, compared one thing with another. I said that there's a term that I used to work in Belfast, and the Good Friday Agreement was that the IRA should put their weapons beyond use. It means they didn't surrender the weapons, but they were documented to have made sure that they could not be used again. So I'll do this. I'm not surrendering my instrument to you, but we'll basically destroy the power supply so it can't be used. And you're happy with that? And they said, yeah, all right, go on, then you can have the discount. So that was another interesting little sideline. Anyway, the interesting thing was we got this paper out. New England Journal is one of the highest, highest impact factor of any journal. Great to celebrate that. But we'd never even met the German people. We said, well, we better come over and have some champagne with you guys. So we went over to Germany, and there we are, having some champagne in Hamburg. And myself and Martin, we said, well, you know, what are we going to do next? People just think we're a one-trick pony, you know, we just got lucky with this. We need to do something else that influences the field. He said, yeah, I'm up for collaboration. I said, well, we've got to think, what are our unique selling points? And he said, well, I said, well, you know, we're quite good at the bioinformatics side of stuff, and, you know, analysing genomes, analysing sequences. He said, I said, what's your unique selling point? He said, I got a freezer full of shit from the outbreak. I didn't put it quite that bluntly, but that was the drift. Okay, why don't we actually just sequence your shit and see what we can discover? Okay, sounds like an idea. And basically what I was tapping into was this thing that in the 21st century, we're still using 19th century techniques. So clinical microbiology, bacteriology, relies on the Gram stain, which was invented in 1884 by Hans Christian Gram, and culturing things on solid medium as colonies, invented by Robert Koch in 1881. We're still doing this. We're still growing things on agar plates and then Gram staining them well over 100, 120, 140 years later. What do we get? You know, is there? So what I said is, look, okay, this is what we've done up till now. You've taken those shit samples, we've plated them out, and then we've sequenced those colonies. Let's cut out the middleman. What if we sequence that shit? Could we actually get the same results, get the same understanding of what's going on in this outbreak, without having to do the culture first? Could we do effectively diagnostic or clinical metagenomics? So they said, yeah, all right, sounds like an interesting, innovative idea. Let's give it a go. And we had no idea what kind of results we were going to get. And I said at the outset, I said to Nick, look, if we sequence a fecal sample and we can detect the sugar toxin genes in those sequences, I'd count that as a win, basically. Okay, we can detect, not by PCR, not by culture, we can detect just by sequencing metagenomic sequences, we can detect the sugar toxin and say, there's sugar toxin in that stool sample, and maybe not in that stool sample. That would be a win. And what coloured my thinking there was an assumption that E. coli is a bit player in the gut microbiome. Okay, so when you talk about, if you ever used to go to talks in those days, you still get it today, is that people would always, when they're talking about the human gut microbiome, they say, the ratio of Firmicutes to Bacteroid ETs, these are the two big phyla that dominated in the human gut. And the E. coli, as remember the proteobacteria, gamma proteobacteria, that's a bit player, less than a percent or less than a percent of the biomass. So my assumption was, so we'll be lucky to find any bits of sequence from E. coli. If we can get basically the sugar toxin, that's a win. So Nick, we did the metagenome sequencing. Nick then did a mapping against the outbreak strain genome. It was one of those, that's half a dozen times in my work, and I've effectively knocked off the chair, like, what? Really? So what we found was we got over 20x coverage of the outbreak strain genome in this metagenome sample alone. And we could see this skew here, and this is what we were talking about. Nabil was telling you earlier about the regions around the origin are overrepresented in the genome. So we saw this funny skew. We saw this funny little uptick around the sugar toxin encoding phage. And we interpreted that to mean that, so there are two kinds of sequence that contribute to the sugar toxin gene sequences in a metagenome. There are those that are derived from the bacterial chromosome, where the bacteriophage is integrated into the chromosome and is at the same copy number as all the other chromosomal genes. But as part of the disease process, those bacteriophages lyse the host cells that the bacterial cells are in and produce many, many phage particles that are released and then go on to infect the next cell and so forth. And so we would expect to see much more of that phage sequence and of the sugar toxin sequence than we would of the chromosomal genes, and that's indeed what we saw there. So we were absolutely astonished about this. And there was such a depth of coverage here that we could actually just run it through the program Torsten Seaman had written to determine MLST profiles from genomes, and it told us it gave us a sequence type profile. So this was, wow, this is, astonishing to see this in a metagenome like that. So we, as it turned out, the Journal of the American Medical Association had a feature on medical genomics that they were going to run. And we said, well, look, we've got this little study we've done. We've done it all on our own mice heat. We've done just a few of these. And we thought this was pretty revolutionary. We made inquiries with the editor, would this be suitable, this kind of thing you're after? And they said, if this is a diagnostic test, it's too few samples, it's underpowered. And what we'd done is we'd taken a bunch of samples that we knew had come from the outbreak, some of which had abundant amounts of the STEC strain, some of which had very small amounts, marginal, and we'd only got sensitivity of 67%. So effectively as a test, it was fairly lousy. And the other vibe that we got back from them was that by aligning these metagenome sequences to the outbreak strain genome, we were cheating. Because in a real outbreak, you would not know in advance necessarily what was causing the outbreak. So for this to be really meaningful, you'd have to be able to recapitulate what the outbreak strain genome looked like just from the metagenomes without using a cheat, without using a crib. I've made the analogy before that this is a bit like trying to solve a jigsaw puzzle by taking the lid of the jigsaw box and aligning everything against the picture you see on the lid. It should be done in a way you don't know what the final picture looks like. So we had to go deeper and we had to go smarter in this. And so this guy, Jeff Smith, was someone we knew at Illumina. We had kind of links to him. And they just released a new instrument called the HiSeq 2500, which provided massive stonking throughput. And we said, look, can we just blag one of your runs on there? And we'll bring you in as a co-author. And it will allow you to sort of, beta test your instrument. It will provide a great test case. And he said, yeah, fine. All right, then we'll do that. This is Nick. In those days, I don't know if Nick blogs so much now. He spends a lot of time on Twitter, but he doesn't blog anywhere near as much. But in those days, he used to blog everything he did, sort of thing. And he was just showing the huge, what was then considered huge throughput from that run, an awesome amount of sequencing we managed to get done. And then we actually looked in more detail at all the samples we had, and this shows you, there was some variability. There were some even more striking examples. So this one here, we were getting over 500x coverage of an E. coli genome just from the fecal metagenome. So to get 500x coverage from colonies that you've grown in the lab is pretty spectacular. To get it out of a metagenome, it was absolutely astonishing. You can see in other cases the coverage wasn't quite so high. In these other two here, it was about 10x. And we're getting this strange uptick. I'm not quite sure why the position changed slightly for the sugar toxin, but we still see the phage going up there. But another thing that we noticed was that in some of these samples that were supposedly from the outbreak, we were seeing other pathogens as well. So we could see Clostridium difficile in the sample. We could see Campylobacter concisus, which at that time still is kind of emerging diarrheal pathogen. People are not quite clear what to make of it. And we saw Campylobacter jejuni. I can't remember, I think that was in one that they gave us a number of samples from patients with diarrhea that weren't in the outbreak, where they knew that there were other causes. And I think this was one where we could show that, you could realign, you could align the reads against that from a metagenome and see coverage as well. So this just it tabulates the same kind of thing here, that we're getting C. diff, we've got Salmonella enterica, Campylobacter jejuni, and we could actually get useful information from the sequences in the metagenome. So we could say, this C. diff has got toxin A and toxin B. We could actually say that this Salmonella enterica, we could actually serotype it, and we could also with Campylobacter jejuni, we could detect the various toxins that were present. But when we did it across the whole, much larger data set, we were still coming up with only 67% sensitivity. So if we're trying to say, metagenomics is a wonderful new diagnostic test, people say, you've only got 67% sensitivity. That's a bit rubbish, isn't it? So we said, well, what's the question? Let's see if we can change the question. We're not going to sell this as a diagnostic test. We're going to say, let's see if we could use metagenomics as a way of identifying the cause of an outbreak a priori, with no prior knowledge of what might be causing the outbreak. Would it be possible to do that? So we had to go away and think about this, and we recruited in this guy, who some of you might recognise in his younger form. This is Chris Quince, minus 11 years, looking rather younger and fresh-faced. And so we had discussions with him about how he would address this issue. And Nick actually did the analyses, but with Chris helping a great deal in this. And what Chris suggested was, let's say that we take all of these are effectively contigs, and we've got this depth of coverage, and we've got the GC content there. And if we sort of look at it like that, we can see that different kinds of bacteria are in this kind of space, all overlapping. There's a very complex community there. How can we simplify that? So the first thing that we did was we said, well, we don't want things that are just present in one or two samples. What we want are sequences that are present in most of the samples. I think we had 40 odd samples, so we want at least half of those outbreak samples. We want sequences that are in those. So if we retain those, what happens? And we get rid of all the things that are only present in one or two samples, or three or four. And you can see you do get a massive simplification of the population there. It becomes a lot easier. And that took us some way forward, but it still didn't solve the problem. So anyone got an idea how you might solve the problem? And this is where, Chris and Nick, they got very agile minds, and they could see, that's how we solve the issue. wonderful. There we are. You're gone to the top of the class again. So that's exactly what they said. Right, look, we've got all these reads available in the public domain for normal people. Why don't we just subtract everything that's seen in that normal microbiome from this and see what's left? And that's what they did. And they ended up, when they did the taxonomic classification on that, you can see that they managed to get not quite exclusively, but very, very concentrated data set from the Enterobacteriales, which is in fact from the outbreak strain. And in fact, they then managed to sort of reconstruct the outbreak strain genome from this kind of approach, and they could show that they could detect the sugar toxin and everything else in these samples. And they wrote that up as a paper, again, which not quite as highly cited as the one on the benchtop comparison, but one of my most highly cited papers. And there's a guy, David Relman, who we'll be talking about this afternoon a bit more, who's one of the big guys in the field of molecular microbiology, microgenomics in the US. It's based at Stanford. And we got him over to give a talk a little while after this, and we said, we think you were reviewing this, weren't you? And you were the one telling them how they should guide us. And he said, I might be. And he said that he took his hat off to. He said that in the US they'd spent, I think, certainly tens of millions, maybe hundreds of millions of dollars on the human gut microbiome project. They spent a huge amount of effort getting all these labs to standardise their approaches, to analyse it. He said, you're the first person just to say, why don't we just see what happens in disease when someone gets infected? You know, everyone had spent all their time worrying about how do they get the best possible picture of the gut microbiome in health, and we were the first to say, why don't we just use these approaches in this way? So that's me finished for the introductory talk. So, yeah, that short period of just a few months where we broke a lot of paradigms, both infection itself did, and we did when we did stuff in the labs and elsewhere. So, you know, infection still, we know it's a threat to advanced societies. We cannot say that infection's over. We had that diarrheal disease. We've not seen anything quite like that since then, really, in terms of diarrheal outbreaks. But, you know, you've seen COVID. We have still the ongoing threat of antimicrobial resistance. Hasn't gone away. So you're all going to be in a job for the next 10, 20, 30, 40 years. We can be certain of that. Basically, bacterial genome sequencing, in this case, you know, they did the sequencing in a few hours and they released it. And that was, again, a game changer, like, boom, off it goes. There's a sequence out there. Serious science, you know, well, you can do serious science using social media. Here's an example of a man he's a social media human symbiont, because he's plugged in to social media, and he cannot live unless he posts how many times a day? 8 times a day? 10 times? 4 times?
I don't post that much.
Yeah.
Once or twice a day, that's it.
This is the sort of the mild version. The major version, people like Nick Lohmer and Biomick Watson, you know. I went to see Biomick Watson. That's his name he uses on Twitter. His real name is Mick Watson. I went to see him up in Edinburgh, and he's so obsessed. He's having a conversation with you, and he's looking at his phone and tweeting as he's talking to you. You know, that's one extreme, but there is serious work to be done on social media with blogging and everything else. Pathogens fall into tidy pathotypes. Well, yeah, maybe they do, maybe they don't. They don't really, and you have to be careful about that. We weren't the first to do clinical metagenomics. It had been done on a few viruses before then. Still only a handful of papers before we did it, and there was one Japanese man who sequenced the stool samples from someone with Campylobacter and detected a few hundred reads from Campylobacter. So we weren't quite the first to do it as a diagnostic approach for bacteria, but we did change the landscape, and that work continues. So Justin O'Grady here, who now works for Nanopore, has been one of the pioneers taking that seriously forward to say, can you use a metagenomic approach where you don't have to wait around to diagnose the pathogen by culturing it? That's still going on. And the interesting thing that we saw, I didn't say much more about it, but we did see in some of those patients who clearly had the EHEC infection, they had, say, Clostridium difficile in an appreciable, detectable amount in their stool. And that raises the question, what was the C. diff doing? Was the C. diff simply hitching a ride? The conditions in the gut changed to favour it, but it wasn't actually contributing to the progression of the disease. Or was it perhaps actually also contributing to the disease? And was it a cofactor in the disease? And that's an interesting question that hasn't been addressed, but it is one of those things that once we get beyond the constraints of picking colonies, growing things on plates, and actually looking in the round at what's going on in the microbial communities, we are going to get more sophisticated insights into how that disease process kicks off, how it's maintained, and how it's resolved over time. And that's it. That's me. I get a bit waxing a bit lyrical. There's me trying to knock down the Berlin Wall as a metaphor for the way in which these assumptions come falling down. If you want a more kind of quote with gravitas, there's this one here from Alfred North Whitehead, who was a philosopher, who's he said that the aim of science is to look for simple explanations, and we think that we fall into this trap of thinking that the facts are simple because we've got a simple explanation. But the guiding motto should be seek simplicity but distrust it. And this is one of those things where the educationalists tell us that the goal of any educational event like this is to transmit facts, to provide people with skills. But the third thing that we try and do is change attitude. So I'm hoping to change your attitude to the future here, that you're prepared to question assumptions and not just fall in line with what's the simplest explanation. Of course, there's a whole philosophy of science discussion to be had there, that if once you abandon the idea that the simplest explanation is the best explanation, you open yourself up to conspiracy theories, which is another thing that you'll be talking and learning about later in one of those master classes. We'll be looking at how do you deal with anti-science, anti-fact, conspiracy theory stuff. But that's for another day, and that's me finished. Good.